All right. Let's see. So this upcoming week, you guys are gonna be doing the field trip. I guess just the mini like walk around campus. So we're not gonna have time to do review. So once again, I'm gonna do two videos, one covering gymnosperms and the other covering angiosperms, just reviewing all the stuff we went over previously in lab. And the focus of what I want to talk about is simply like life cycle stuff. So life cycle and reproduction of the gymnosperms and angiosperms, all the seeded plants. I'm not really going to get into too much biology. That uh, you guys already know how to study for the biological facts of these kinds of things, right? You've done the lab practical. You know, like each taxa in the slides have like one or two or three main facts that you kind of got to memorize. And those are kind of recapitulated in the lab notebook as well. So you should know by now what is important. I think the most difficult stuff that you guys will have to encounter, and I think the bulk of the problems that will be on the lab practical will have to do with identifying different parts of the reproduction and life cycle of these plants. So that's really what I want to focus on. And really, those are the subjects that have the most moving parts that you really will potentially be confused by. Okay. Um, and I think how I want to start this. So we're going to go over gymnosperms first, right? Gymnosperms. You know, all the conifers and their relatives. But before I get into that, I would just like to really quickly go over fern reproduction from the top, just because a lot of the things that you gotta identify, you know, the sporangia, the antheridia, so on and so forth. These structures are really obvious and simplified in the fern life cycle, and when you go into like the angiosperms and the gymnosperms, some of these structures can be a little bit more derived and harder to spot. So I'm just gonna go over the ferns real quickly so you can track those pieces and then kind of relate that to the more advanced plants. And in case this is still a problem area for you, I did send out that spreadsheet with all these different parts and where they're found in all these different plants. So definitely look at that if you need to have a reference point for all these things. Okay, so let's do the ferns. Ferns for reference. So right? Like all the vascular plants, their main plant is the sporophyte, the 2N sporophyte. Right? In the on the bottom parts of the leaves of the fronds of the fern, you guys have there's sori, right? And the sori, they're collections of spores, or sorry, they're collections of spores. And these collections of spores are called the sporangium, right? Remember, angium, and then whatever comes before that. This is a collection or an organ that makes and stores whatever this blank is, right? So it's a gametangium, stores and makes gametes. Sporangium stores and makes spores, right? And so the sporangium is this organ that contains all these spores inside. The spores, they come out, remember, spores are 1N and the sporophyte is 2N. So how do they make this spore? They undergo meiosis. And this specific part of meiosis wasn't really talked about or emphasized so much in the fern lectures, but right, there has to be a mother cell that undergoes meiosis to make the spores, right? And that mother cell is called a sporocyte. Not a sporophyte, but a sporocyte. I realize it sounds very similar. It can be very confusing, all these terms, but it'll help maybe to remember. So phyte is a suffix that refers to plants, right? So the sporophyte is the spore-making plant. The sporocyte, site refers to cell. This is the spore-making cell. So this is a 2N cell. It's just a body cell. It's a modified body cell of the 2N sporophyte, right? And it doubles its DNA, and then it splits off into 4 individual cells, which is the whole process of meiosis, to make 4x1n spores. That's the whole process of making spores. So there are a couple of different names for this. Sporocyte or spore mother cell, which is also encountered in your literature that you have to read. So just know that these are interchangeable. And I'm just drill this through your head. The sporocyte is the mother cell that undergoes meiosis to actually make the spores, right? And so now you have these fern spores. The spores, they germinate, and what comes out is the gametophyte, right? This little thing in here grows up through mitosis and becomes a gametophyte. And in the ferns, the gametophyte is its own separate plant that lives separately, and at least in the simplified version that we went over in class, the gametophyte is bisexual. And we'll have eggs stored in the archegonia and the sperm down here in the antheridia. The sperm get released, they're motile, they swim around, they'll find another gametophyte which has eggs stored in its antheridia. The sperm will find that egg, fertilize it, turn into a 2N zygote, which grows on, like, within the antheridia, sorry, the archegonia, and it'll sprout out and make a whole new sporophyte. And that is the simplified version of the life cycle of a fern. Now, all these moving parts, the sporophyte, the gametangia, all these different things, are also present to some degree in the seeded plants. But a lot of these traits can be derived and very, very reduced. And that all has to do with that increasing march towards terrestriality, right? The most advanced plants, angiosperms and the gymnosperms, they are much better adapted towards living on dry land than the ferns, and I'll show you how that works. So let's, without further ado, let's get into the gymnosperms. I'll get to a new page because there's a lot of stuff in here. So again, these are the conifers and relatives. So like pine trees, right? So let's start from the sporophyte stage. You have the pine tree. It's 2N sporophyte. And if you recall, on the pine tree, you have these, on the tips of some of these branches, you'll have cones or strobili. So you have the female strobili, which is big, and the male strobili, which are small and grouped together. And so remember, for future reference, everything that has to do with female related organs have the prefix mega, and then micro for all the male. Okay, and I think that just has to do with the size of the gametes. That's why they call it mega versus micro. Okay, so this is the mega strobilus right here. Strobilus. It's the micro strobilus. And let's take a look at the female one first. And so if you peel apart like a pine cone, a female pine cone, a megastrobilus, you take one of those scales, it looks like this. And you've seen this in your lab notebook, right? Each one of these is an ovule. And we went over this in class a bunch of times. Just remember that an ovule will turn into a seed. Everything that is an ovule eventually turns into a seed. Okay? And so each scale, which is a modified leaf on one of these pine cones, it contains 2 ovules. So what we're going to do now is we're going to take a cross section of this pine cone scale, this female one down this way. So maybe 3D, it'll look better like this. Cut down like that. And what you get is something that looks like this. And there's all these different terminologies, like this is the bract and this is the. I'm not going to go into all those details. I would just want to note that this entire thing right here, this is a modified leaf. And you've already seen this terminology before, but this is a sporophyll. Right. So now we have three terminologies. We've got the sporophyte, which is the phyte refers to the plant, so this is the 2N plant. You got the sporocyte, which is the precursor to the spores, the spore mother cell, cyte, cell. And then phyll, I think that means leaf. So this is sporophyll. This is the spore-containing leaf, and that's, if you remember in the strobili in the seedless vascular plants, that's exactly what they are, right? They're these leaves that at the base of the leaf contain a collection of spores. Well, right now at this exact stage, this leaf does not contain a spore, but this structure right here, this ovule, actually on the inside, it's an organ that will eventually make spores. So this is actually considered the sporangium. This enclosure here. And this is the sporophyll, this is the leaf itself, right? This is all 2N tissue, right? This is all coming from the mother sporocyte. So inside the sporangium right now, before any spores are made, you have the megasporocyte. Megasporocyte. The megaspore mother cell. Okay, and this is still 2N, right? This is still a cell of the sporophyte. Okay, then it undergoes meiosis. And what results is now you have 4 single cells. And remember, what comes from the meiosis of the sporocyte is spores. So these are spores. These are megaspores. Unlike the spores in these seedless vascular plants, these spores they don't have a hard shell outside of them because they never actually leave the sporangium. Okay, so whereas the ferns, they take those spores and they fling them like really far, and they maybe travel in the wind or in the water, and they find a place and they germinate into a gametophyte, which is what comes out of a spore. These spores don't need that shell because they never leave the actual sporangium. Instead, what happens is, so 3 in the female plant, at least 3 of these. They're not the female plant, the female cone. 3 of these spores, they abort. So what you're left with is 1 remaining megaspore. And remember, what comes out of a spore, what germinates from a spore in the ferns, is a gametophyte, right? In this case, the spores don't actually germinate because they don't have to sprout out of a hard shell. Instead, the 1N spore, the megaspore, just develops through mitosis, into a female gametophyte. This is the megagametophyte. And there are other structures surrounding or inside the sporangium, like the nucellus and like the integument. Is that what it is? Integument, yeah. That you will have to memorize, but are not intimately important in this whole process, so I won't really go into detail. I don't want to confuse you. I'll have these on notes later, so just bear with me. This is just to map out the different features of this reproductive cycle, so don't fret about that too much. Anyways, so this is the megagametophyte. It came from the mitosis of this megaspore, right? The spore doesn't germinate, no germ, instead simply just mitosis. So normally, like in the fern, it germinates, and what comes out of it, the baby gametophyte undergoes a bunch of mitosis and forms an actual gametophyte. In this case, it just skips the germination stage. Okay? And so in the megagametophyte, you will have an egg. Or in the slides that we saw at least one egg, usually two eggs. But only one gets fertilized. And if you remember the gametangia, that's the organ that houses and makes the gametes. In this case, in the female, it's the egg, right? So basically, this little pouch that is holding the egg is the gametangia. In all plants, gymnosperms, and earlier, the ones that we've gone over, just the receptacle, wherever you see the egg in that plant, the receptacle that's holding that egg is the archegonia, or the female gametangia. So that's the archegonia. These are the two archegonia that hold the two eggs of the megagametophyte. And so we're going to leave this female stage right here for now. We're not going to go any further until we fully describe the male cycle. And then we can get the two gametes together and make a zygote. So in the males, it's very similar. So you take one of the scales from male strobilus, and because the male doesn't have any ovules, the seed, or sorry, the scale of the strobilus just contains two sporangium. So each one of these is a sporangium. If you cut it open like this, look on the inside, you see a lot of, in the immature one, you see a lot of cells like this, these big cells. These are microsporocytes. So these are the mother cells of the male spores. And like the female spores, like the female sporocytes, they undergo meiosis. They make these tetrads of cells. So these four cells stuck together. Unlike the female cells, so these are the spores that are stuck together. Three of them do not abort. All of them stay alive. And these are all one end, right? So that's different from the female spores. And each one of these spores will mature into a pollen grain. And that pollen grain is basically a male gametophyte. So this is a pollen grain. Inside the pollen grain are two cells, the germinative cell and the pollen tube cell. I think in the pine trees, at least, so this cell is going to eventually turn into sperm. And I think they are technically stored in a very, very reduced antheridia. But let me double check that. I'm not going to comment definitively one way or another. But if they are housed in antheridia, which is where you would house and make male gametes, the sperm, then it's greatly reduced because the entirety of the male gametophyte, of the pollen, male gametophyte equals pollen grain. It's basically consistent of two cells in a capsule. And then the Mickey Mouse ears, these are filled with gas to allow them to float in the air really well. Okay, and so this male gametophyte will then find its way to the base of a scale on a female pine cone, and it will then, once it senses it's near that female gametophyte, what it does is this cell, generative cell, turns into sperm, and then the pollen tube cell undergoes mitosis and just makes a pollen tube, which will find its way to the actual egg, and then the sperm that are made from the generative cell go through this pollen tube, and one of them will fertilize this egg. And what results from this is an embryo. And the embryo is, let me backtrack. What results from this fertilization of the egg is a zygote, right? And one of the two eggs, or one of the multiple eggs, gets fertilized. Only one. And through mitosis, that 2N zygote becomes an embryo. And I'm not exactly sure how this happens, but the rest of the body, so this embryo is made inside the archegonia, right, where the egg is held. That's where the sperm will fertilize the egg, and that's where the zygote is created, and it will undergo mitosis to form the embryo. So it's living inside of the gametophyte, right? That gametophyte, that female gametophyte, which is one end, her tissue somehow becomes endosporic material. So it becomes the endospore of the seed. So this is the embryo. And remember, the endospore is the nutritive material. So it provides nutrients for the embryo once that seed, falls onto the ground and starts to germinate. So going backwards from the outside in, this 2N out here, this is the sporophyll. This is all made out of 2N material from the adult sporophyte. All this stuff here, all the endospore is derived from the archegonia, or derived from the megagametophyte, which resulted from the meiosis of the megasporocyte. And then the embryo inside is 2N. It's a different 2N than the 2N here, right? Because it's the 2N of the fusion of a male and a female gamete. So it's got half the same DNA as this guy here on the outside. That makes sense? Great. And eventually, right, each scale makes 2 of these seeds. So all this stuff is in here. This embryo, right, and this is the wing of the seed. And they'll fall out, and they'll catch the wind with their wings, and they'll fly to another place, and they'll germinate into that's ugly, into a new sporocyte, or sporophyte. And the cycle continues. A few things to note. Just know from what I said, remember where the gametophytes are, the gametangia, the spores. Just to review from the top, I guess, remember that the spores never leave the sporangia before they turn into a fully mature gametophyte. Whereas the ferns, they spit out their spores. These spores stay inside the parent sporangium, and they will develop into the gametophyte inside the sporangium. In the case of the megagametophyte, the megagametophyte, the female gametophyte never leaves the sporangium ever. Whereas the male gametophyte, the pollen grain, eventually will leave, but only after that spore has turned into a megagametophyte. Other things to remember always the archegonium is that the pouch inside the gametophyte, the female gametophyte, that houses the egg and makes the egg. So wherever you see the egg, that pouch that holds the egg is the archegonium. In the antheridium for the pollen, which is the male gametophyte, it is either absent or very greatly reduced. I don't think there's much else to add there. If I think of anything, I'll send you guys an email. But I think I covered that pretty thoroughly, just in terms of front to back, what you got to know for the life cycle, very simply. Again, if you have any problems tracking the different cell types, the different structures, please refer to that spreadsheet. I know it's pretty long and a little bit complicated, but again, if you have any questions, just come to me with questions via email or via office hours. This week, I will be down in San Diego teaching a workshop. So if you have any questions and you want to have like an actual face-to-face interaction, I will be available through Skype. Anytime you need help, just email me, and then we can set up a Skype interview, and then we can talk about all the things. The Skype now I think has this capability we can actually write. I can show you what I'm writing through some digital blackboard, and it'll be just like office hours. Okay, so next I'll go into the angiosperms, which are the flowering plants. So I'm going to stop this recording now. 1, 2, 3, stop.